लिंकेज एंड क्रॉसिंग ओवर। क्रॉसिंग ओवर के बारे में तो आपने पहले भी पढ़ा होगा। क्रॉसिंग ओवर that is an event that is going to occur in case of meiosis, that is a packeting, right? But how crossing over occur, what are the roles of linkage during the process of crossing over, and how it leads to the failure of Mendelian law of independent assortment, that is we are going to learn today. This is Dr. Pankaj Kumar, your mentor for this particular sections. So today we will be discussing about crossing over, linkage as well as the linkage map, right? First of all, what is crossing over? I've already told you. So this is the event that is going to occur during the meiosis, right? And in case of meiosis, when the homologous chromosomes come very close to each other, right? Then between the non-sister chromatids, the chromatids get exchanged, and that is going to occur during the event of packeting. Okay. So the sequence of events leading to crossing over, we can say like this: that pairing of homologous chromosomes in zygotine, that is what we call synapsis. Then it is going to happen like that. Then it will go to the tetrad stage, right? And when the tetrad formation is going to occur in case of packeting, then this is the place where actually the crossing over between non-sister chromatids is going to happen. And the site of crossing over, what we call is the chiasmata, right? Now, what are the steps? You can see there. This is how the chiasmata has been formed, right? And you can see there that the portion actually get exchanged over there, right? So this is the new recombinants, we can say, okay? And finally, we can say as a result of gametogenesis, we are going to have two parental one and the two recombinants one we are going to be formed, right? That is a normal process what happens. So percentage of chiasma, we can always say that is 2 into percentage of possible products. So that is going to occur. Percentage of chiasma is equal to 2 into percentage of possible product. That's happening. Now, what are the mechanism of the crossing over? How it is actually involved? You see, over the period of time, number of theories has been given over there, right? First thing first, the crossing over always occurs at the 4-strand stage. Why 4-strand stage? Because out of 4, 2 participate in the crossing over. So the maximum possible recombinance is only 50%. Beyond 50% is not possible. So that's why the crossing over is going to occur only in the 4-strand stage. Okay. So crossing over is nothing but it is actual physically breakage and reunion process. So breakage is going to occur at the part where actually it is going to cross over, right? And rejoining occurs between non-sister chromatids during the physical exchange of the chromosome. Okay. So as a result of physical exchange, what will happen? Recombinants are going to be formed, and this mechanism has been explained by the Holiday model. Okay, the Holiday and Whitehouse model that very clearly explain the entire mechanism. And Mendelssohn and Wiegel demonstrated that recombination involves breakage and reunion. So this is a very brief introduction about the crossing over. But the main thing of today's session is that we have to discuss about linkage. Now, what is linkage? You see, there are certain genes that have a tendency to inherit together. Okay, so linkage is a tendency to inherit together, right? And as a result of this, what will happen? That law of independent assortment is going to fail, right? And the equal proportion of gametes is not going to be formed. Okay, so the tendency of the gene to inherit together is actually known as linkage. Why does it happen? Because the genes are very close to each other. And since genes are very close to each other, it is not possible to have a crossing over between these two genes. So that is why it leads to linkage. Okay. So, little bit about history. Linkage, first of all, it was reported in a sweet pea by Bateson and Punnett. And later on, the mechanism of linkage was explored by Morgan. Okay. So in today's sessions, we are going to see both experiments. What Bateson and Punnett has done, what were their observations, right? And where they were inconclusive, they explained the event. Unfortunately, they could not explain the phenomena of linkage. Later on, by the experiment of Morgan, we came to know the very phenomena of linkage. Okay? So first thing, let's talk about Bateson and Punnett experiment. Very interestingly, you see, Bateson and Punnett were actually trying to redo what Mendel did way back in 1856. For a change, they have, instead of garden pea, they have taken a sweet pea. Okay, and again, a typical dihybrid inheritance where they were studying, and for this, they have taken the flower color and the pollen same. So, in case of flower color, in case of the sweet pea is a purple, that is the dominant one, and red was a recessive one. Similarly, the long pollen was a dominant one, and round pollen was a recessive one. So, the pure plants were taken, and as expected, in the F1, they are going to get purple long, because both were dominant, right? So, in F2, it was gone for the usual self-pollination, and is a result of self-pollination. Very interestingly, they were expecting this ratio, 9:3:3:1, typical Mendelian dihybrid ratio, dihybrid phenotypic ratio. But instead of that, what they got 7:1:1:7. So mind it, here the parental combinations are more, recombinants are less. Okay, this was the thing what they observed, but they could not explain it. What could be the reason, right? So what they observed, that Mendel's law of independent assortment is not operating. Had it been operating, they would have gone 9:3:3:1 ratio. Second observation was that the parental forms are much more than the recombinants. Okay, and the number of recombinants are very less. Okay, so as I told you, Bateson and Punnett failed to give the explanation for Mendel's experiment, right? But they simply say that there is a tendency of these two characters to inherit together, but the real explanation for the linkage was actually provided by Morgan while working on Drosophilla. Okay, so let's have a very detailed study of Morgan experiment. So in order to understand the Morgan experiment, first of all, we need to understand the characters, what actually he took. Morgan studied two different crosses involving genes that was placed on second chromosome of Drosophilla, right? What was the character they were taking? The long wing is a wild character or natural character, right? And is a dominant, and it was a dominant over recessive allele, that is a short wings. And similarly, gray body, that was again a dominant character, and it is recessive over the black body. Okay, so these two characters were taken by Morgan, right? And what Morgan did? Morgan, first of all, they crossed long wing gray bodies to short wing black bodies. Okay, so you see, long wing and gray bodies. So they were the dominant one, short wings and black body, they were the recessive one. Okay, so I've just shown the genes that is written over there, right? So this was the cross happened, and this is the gamete that is going to be formed. So in F1, they are going to get long wing and gray bodies. As usual, both the dominant characters are appearing, so that's why they are getting long wing and gray body. Okay. So the F1 genotype was like that. This was the F1 phenotype, and F1 genotype we can say like this. That is, this is a complete heterozygous, because we know very well that in F1 we are going to have a heterozygous composition. Okay. Now what happens in the next series of reaction? This F1 heterozygotes were test cross, and we know very well, if Mendelian experiment or Mendelian law of independent assortment is operating, then in case of test cross we are going to ratio of 1:1:1:1, right? So this is the ratio what we are going to get in case of test cross. Now have a look what actually they got. Okay, so what they got when they did the test cross, what they got? Long wing gray bodies 41%, short wing black bodies 41%. So the parental combinations were much more, right? And the recombinants, that is long wing black bodies, just 9%, and similarly the short wing gray bodies just 9%. So you can see there, right? So how the difference is going to happen. Now in the second series, what Mendel did, what Morgan did, that they have taken a long wing black bodies and short wing gray bodies. Okay, so one dominant, one recessive. Okay, in a cohort. So the genotype is like this, you can see there. So this is the genotype, right? So in the next what Morgan did, he took long wing and black bodies. Okay, so in case of long wing black bodies, what happens? There is a one dominant gene and one recessive gene. You can see there. So this is a dominant one and this is the recessive one. Okay. And as a result, you can see this combinations, right? And finally, we get long wing gray bodies, like because long was a dominant and gray was dominant, so F1, the phenotype remains the same. But their position of genes were entirely different. So in one chromosome, they are going to have a one dominant, one recessive. In another chromosome, they are going to have a one recessive, one dominant, like this. Okay. So just see that in the previous case, what happens, both dominant were on the same chromosome, both recessive were on the same chromosome. But here it is not the case, right? So again, such variety were put to a test cross. Now, as a result of test cross, what happens? That parental combinations, now long wing black bodies, because this was the parent from where actually we have started, right? Again it got 41%. Short wing and gray body, again it got 41%, because you see, we have started with a long wing, that is a one dominant and one recessive. So black bodies was a recessive character. So here we got 14%. Here also, this was a recessive. Short wing and gray was a dominant, right? So we get 41%. But the recombinants, because the new combination, right? That is the long wing gray bodies and the short wing and black bodies, both are going to get 9%. So this experiment, the two test cross, was just an eye opener. Why it is an eye opener? Because you see, it was very clear that the genes which are inheriting from the very beginning, they tend to inherit together, right? They do not tend to cross to each other. Okay, this is all about the linkage. So what was Morgan's observations? That in cross 1, F1 carries the wild type form on the same homology, right? And this element is called as a coupling or cis configuration. That means whenever in a chromosome, both dominant gene are present in one and both recessive are present in other, this is what we call coupling, right? So as you can see there, so both dominant is here and both recessive is there. This is what we call coupling one, right? Now in a cross second, what we see that there is a one dominant, one recessive, one dominant, one recessive, one recessive, one dominant. If this is the condition, we call it is a trans configurations. Okay, so these two configurations came into existence, right? And as a result, what Morgan concluded, that regardless of the fact whether the cross is done in cis or trans configurations, if the genes are present together, they always inherit together. So that is why in case of trans, what we have seen, that the long as well as the gray, they were inheriting together, so that's why they are 41%. But in case of trans, what we see, the long were associated with the black, right? And they were inheriting together, right? And in that case, again, they are going to produce 41%. So one thing is very clear, that if the genes are linked together or together, they always tend to inherit together, right? So these are the problems that is asked in your NEET examinations. Okay, so a test cross of F1 was crossed with plus plus AB. Mind it, this plus plus is showing the wild type. Okay, so what we have to figure it out, how you can say whether it is in cis configuration or trans configurations. Okay, one thing is very clear, while seeing at the entire things, what you have to figure it out, that which number is more. So number which is more is obviously it is a parental type. Okay, so here you can see there, so here you can see there that these are the parental type. Why? Because this number is absolutely more, right? Now this plus and this A, that means it is going to be arranged like this. Okay, so that means if I take another homology, okay, so this is going to be B and this is going to be plus, like this. So one dominant, one recessive. Okay, so that's why it is going to be absolutely in trans configurations, right? Now let's talk about linkage. What exactly linkage? Right now we have seen the phenomena of linkage. Okay, now linkage can be of two types, what we call complete linkage or incomplete linkage. So whenever there is going to be complete linkage, no crossing over, so there is a no chance of formation of recombinants, right? But whenever there is an incomplete linkage, what happens? Failure of law of independent assortment, but crossing over do occur. Very less, but crossing over do occur, right? And very less number of recombinants also. Okay, so this is how we can differentiate between complete linkage or incomplete linkage. Now, just see. I have just taken a very one simple cross. That is a typical Mendelian cross, right? So as a result of cross, we will in the first we will get F1. Okay. In case of F1, this capital A, capital B, small b gametes will be formed, right? So you see, if A and B are very close to each other, what will happen? There is a no crossing over. Okay. And since there is no crossing over, only parental types are formed, recombinants are not formed. Okay. This is what we call complete linkage. Similarly, if I talk about incomplete linkage, same thing, right? So what happens? Suppose there is a 20% recombination. What happens? That there is a few cases of recombinants. Okay. So if there is a few cases of recombinants, what will happen? That 4 type of gametes will be formed. Capital A, capital B, capital A, small b, capital A, small b, and small a, capital B, right? Now out of these 4, what we see, the 2 are the parental ones and the 2 are the recombinants one. Since there is only 20% recombinants, so more parental combinations, right? So that's why what we see that here A, B are not very close to each other, right? Crossing over does occurs, but there is a failure of law of independent assortment. So that's why less number of recombinants and more parental forms are formed. So if I compare incomplete linkage with independent assortment, we can say that the same thing up till this stage, you can see the same thing, right? Okay. So here also the crossing over is going to occur. So what we see that gene A and B are linked, right? So more parental forms, less recombinant. In case of independent assortment, A and B are not linked. So 25% of each gametes are formed. That means 50% parental and 50% recombinants. So in case of incomplete linkage, there is always a failure of law of independent assortment. That is the key learning what we have. Okay. So as I told you, there will be equal proportion of gametes are formed in independent assortment, but there is an unequal in case of the incomplete linkage. Now one last thing, that is gene mapping. Now you see, what is the gene mapping? In case of gene mapping, what we have to know, what is the distance between the 2 genes? Okay, and this is measured in terms of either centimorgan or map unit. Okay, so what happens? Map distance are always directly proportional to the percentage of recombinants, and that is also proportional to the percentage of crossing over. Okay, so if I say that gene A and B show 10% recombinants, it means that gene A and B are 10 map units apart, and we can show like this. So this is a 10 map unit apart. Okay. So we know very well that since the crossing over is going to occur in 4 strand stage, so maximum percent of recombinants will be 50%, right? So the recombination frequency between them cannot exceed the 50%. Okay. So you see there are 2 types of what we can say gene mapping, what we call 2-point test or 3-point test. What is the meaning of point? Point means gene, nothing more than that. If I say 2-point test, what does it mean? That we can calculate only the distance between 2 genes. We cannot know the sequence of the 2 genes. Okay, and it is actually done by doing the test cross. Okay. In case of 3-point test, for a change, here we know the distance as well as the sequence. That is how we can differentiate between 2-point test and 3-point test. So in your syllabus, we have to discuss only 2-point test, not the 3-point test. When you will go to your MBBS college, you will be discussing or you will be learning about the 3-point test as well, right? Let's take one example. So in a particular plant, what happens? Genes for tall, tea plant and red flowers are dominant over dwarf and white flowers. When a hybrid of F1 were test cross, it shows tall red 44, tall white 5, dwarf red 44, dwarf red 7. Now we have to know that what is the distance between gene T and R. That is the question. Now the moment you see this question, the very first question you have to ask, which number is more? The number which is more is by default is a parental one. So here tall red and dwarf white is a parental one. Which is less? The number which is less is the recombinants. So tall white and dwarf red is the recombinants. So if I want to know the distance, so one thing is very clear, that we have to calculate the total number of recombinants. Okay. So you see the total number of recombinants is equal to 12. So gene distance is equal to number of recombinants divided by total number of progeny into 100. So in this question we see that 5 plus 7, that is the number of recombinants, and rest of the total number of progeny. So we got 12 centimorgan. So we can say that the map distance between these two is 12 centimorgan or map unit. So these are the very simple questions that is usually asked in NEET, and I'm sure all of you can do it. Okay, have a look at one more question. Can you calculate the distance between gene A and B? Again, very simple. What we have to do, whatever the number is more, we have to take it as a parental one, right? So recombinant is 18. So altogether we can say that this out of 100, 18 is the number of recombinants. So distance is nothing but 18 centimorgan. So that's pretty simple one, right? Now, these are another type of question that is asked. In a chromosome map, the distance between 4 loci are as follows. Between A and B 10, that is a 10 map unit, B C 4, AD 3, AC 3. Now you have to calculate the crossover frequency between C and D. Actually, you see, this is not a problem of biology. This is a problem. This is a mental problem. This is a problem of mental ability. Okay, just see how we can solve it. What you have to do, you have to go simple by steps. AB, right? So since AB is equal to 10 and BC is equal to 4, so gene C is between A and B, that is clear, right? So this is how we can say, right? Now between A and C is 6 centimeters satisfied, between C and B is 4 centimorgan is satisfied. This is pretty satisfied as per the statement. Now we have to figure it out, the very position of D. Okay. Now you see, there are two possibilities of D. What? D can be between A and C, or D can be beyond A, like this, right? So the correct answer between crossover between C and D is either 9, that is 6 plus 3, or 6 minus 3 is a 3. So that could be the answer. Either it could be 9 or it could be 3. So you have to figure it out that how actually it works, right? So this is all as far as the verification of linkage and crossing.